Please.
Okay, I'm Goodheart. I work on stem cells in the adult brain. So as you may or may not know, there actually exist stem cells in the adult brain which can produce different cell types, including neurons. And through studying these cells, our hope is that eventually we can use these stem cells to replace lost or damaged neurons in diseases such as Parkinson's disease. So part of understanding how these cells work is understanding how different chemicals in the brain interact and have an effect on these cells. I look at the neurotransmitter dopamine and how they impact a subpopulation of stem cells within the adult brain that lies along the lateral wall of the lateral ventricle in the brain, which is this open space in the brain here. So the stem cells reside along here, and one of the main goals of my research is figuring out where dopamine is coming from that supplies these cells. So what we did in this project is we injected a dye into the stem cell zone and then looked to see where that dye traced back to try to figure out what parts of the brain are actually feeding dopamine to these stem cells. So what we found is that the dye actually traced back to an area of the brain that is in between the substantia nigra and the ventral tegmental area of the brain. Both of these areas produce dopamine, but it was previously thought that it is the substantia nigra that actually supplies dopamine to the stem cells. We found that actually it's an area next to the substantia nigra that supplies this dopamine. So that was a really interesting result. So then what we did was we injected a neurotoxin into this area of the brain to see what would happen. We did this in both control mice and also in mice that are missing some of the dopamine in their brain already. And we found that indeed when we injected MPTB, this neurotoxin, there was a decrease in stem cell proliferation in the brain, indicating that dopamine from this area actually does help the stem cells to grow. So that's part of my research. I look at other parts, effects of dopamine, like dopamine receptor presence in the SVC, and I also work with a different neurotoxin. But that's what my poster's on today, so thank you.
Can you talk a little bit about how private support has helped you with your research?
Yeah, definitely. So I was able to spend 10 weeks this summer at 40 hours a week in the laboratory, which was immensely helpful. So when I do research during the academic year, I'm only there for maybe 10 to 20 hours a week, and it's kind of broken up. But by staying there all summer for 40 hours a week, I was able to get a lot done, and I learned a lot too, a lot of helpful techniques. So thank you.